शांतिलाल मेहता बोल रहा हूँ और हम लोग अभी सी सुब्रमण्यम ऑडिटोरियम में 3 दिन की एक जो हमारी संगोष्ठी हुई है उसमें हमारे देश से और बाहर से आए हुए वैज्ञानिकों ने।
अस्सलामु अलैकुम, मैं ज़मीर अहमद अंसारी और आप देख रहे हैं जेड न्यूज़ फॉर यू। अर्ज़ यह है कि आपने अपने चैनल जेड न्यूज़ फॉर यू को अभी तक सब्सक्राइब नहीं किया है तो सब्सक्राइब करें और साथ में दिए हुए बेल आइकन को भी दबाएं, जिससे हमारी आने वाली सभी वीडियोस के नोटिफिकेशन सबसे पहले आपको मिल सकें।
हेलो
जी सर, आपका शुभ नाम?
हाँ, मैं शांतिलाल मेहता बोल रहा हूँ और हम लोग अभी सी सुब्रमण्यम ऑडिटोरियम में 3 दिन की एक संगोष्ठी कर रहे हैं। इस संगोष्ठी का मुख्य उद्देश्य यह है कि किस प्रकार से हम जैविक और जैविक प्रौद्योगिकी के द्वारा हमारी फसलों का उत्पादन कैसे बढ़ा सकते हैं, क्योंकि ये ज़रूरी है कि आज के जनसंख्या के बढ़ते हुए को देखते हुए और कृषि की जो ज़मीन है उस पर दबाव को देखते हुए हमें उत्पादकता और अधिक बढ़ानी पड़ेगी। सबसे बड़ी समस्या आज है कि जल प्रबंधन और दूसरा जो हमारी फसलें हैं उनमें बायोटिक और एबायोटिक स्ट्रेस, जैसे क्लाइमेट चेंज हो गया, जलवायु परिवर्तन से टेम्परेचर बढ़ गया, उससे पैदावार भी घट जाती है। कुछ फसलों के कीड़े मकोड़े और पौधों के रोग, इनमें भी बदलाव आता है। इन सब बदलाव को देखते हुए हम ये चाहते हैं कि जो नई किस्में निकले हैं उनमें प्रतिरोधकता हो, ताकि किसानों को किसी तरह की केमिकल्स छिड़कने न पड़े। और ये आज हमारे लिए ये संभव है क्योंकि जो नई टेक्निक आई हैं पिछले 5 से 10 सालों में, उन टेक्निक से हमारे वैज्ञानिकों के सामने नए हथियार मिल गए हैं, नए तरीके मिल गए हैं, नई तकनीक मिल गई है, जिसके द्वारा हम इन समस्याओं का निदान कर सकते हैं। लेकिन सबसे बड़ी ज़रूरत ये है कि हमें ये समझना पड़ता है कि ये प्रतिरोधकता कैसे आती है। एक बार ये चीज़ का पता लग जाए तो उसका निदान बहुत आसान है। इसके लिए पहले हम लोग जो एनालिसिस करते थे उसमें बहुत टाइम लगता था, कई महीने लग जाते थे पौधों के अंदर उनके केमिकल्स को देखने के लिए। पर आज जो नई टेक्नोलॉजी आई है उससे महीनों का काम दिनों में होता है, तो ये संभव है। लेकिन इसके साथ ही जो नए इक्विपमेंट आए हैं उनके लिए हमें संसाधन भी जुड़ाने पड़ेंगे, और संसाधन के अलावा ये बहुत ज़रूरी है कि जो नई तकनीक है वो सभी वैज्ञानिकों तक पहुँचे। तो हमने ये 3 दिन की संगोष्ठी इसलिए की है ताकि देश से और विदेश से जाने माने वैज्ञानिक यहाँ आए, उन्हें हमें बुलाया है और वे यहाँ आकर के एक अपना प्रपत्र भी पेश करेंगे और जो भी उन्होंने शोध किया है उसके बारे में जानकारी देंगे। साथ ही हमने ये सुनिश्चित किया है कि हमारे जो युवा वैज्ञानिक हैं और साथ ही जो विद्यार्थी हैं, जो भी स्कॉलर्स हैं, पीएचडी कर रहे हैं, एमएससी कर रहे हैं, वो भी यहाँ आएँ और ये जितने सीनियर वैज्ञानिक हैं, वरिष्ठ वैज्ञानिक हैं, उनके साथ बैठ करके चर्चा करें और उनसे नई तकनीक के बारे में जानें। हमने ख़ास तौर से सेशन कुछ ऐसे रखे हैं कि जैसे हमारी समस्या है कि दलहन और तिलहन दोनों की उपज हमारी कम है। आज हमें इंपोर्ट करना पड़ रहा है, बाहर से आयात करते हैं, तो आयात करते में आप सबको पता है कि विदेशी मुद्रा भी लगती है। तो क्यों ना हम ऐसा साधन करें कि उत्पादकता बढ़ जाए और हमें आयात न करना पड़े। तो इसके लिए अलग से सेशन रखा है। साथ ही अब आने वाले समय में और ख़ास तौर से कोविड आने के बाद अब एक हमारे स्वास्थ्य के बारे में जागरूकता बढ़ी है कि जब तक हमारा स्वास्थ्य अच्छा नहीं होगा तब तक दूसरे काम नहीं हो सकते। उसके लिए ज़रूरी है कि पोषक तत्व पूरे मिलें। आज यह संभव हो रहा है कि हमारी जो नई किस्में बने उसमें पोषक तत्व भी ज़्यादा हो। अभी हाल ही में आपने देखा होगा कि हमारे माननीय प्रधानमंत्री जी ने हमारे करीबन 106 पोषक तत्व युक्त वैरायटीज़ का अनावरण किया और हमारे वैज्ञानिक लगातार सतत प्रयास कर रहे हैं कि जो वैरायटी हमारी हो उनमें आयरन ज़्यादा हो, कैल्शियम ज़्यादा हो, ताकि जो एनीमिया की प्रॉब्लम है, जो बोन के ऑस्टियोपोरोसिस की बीमारी हो, वो ना हो। तो कोशिश वैज्ञानिकों की हो रही है कि हम उनको ऐसा अनाज दें जिसमें पोषक तत्व हों और उनको अलग से कोई मेडिसिन ना लेना पड़े। और जो ख़ास तौर से भारत में ज़्यादा बीमारी के रूप में है, एनीमिया बच्चों में बहुत ज़्यादा है, औरतों में ज़्यादा है, कैल्शियम की डेफिशिएंसी भी बहुत ज़्यादा है। तो मैं सोचता हूँ कि भविष्य में जितनी भी वैरायटी होंगी वो कैल्शियम के भरपूर होंगी। साथ ही आपने इस साल ये भी देखा होगा कि अंतर्राष्ट्रीय वर्ष मिलेट्स पे बनाया गया, माइनर मिलेट्स पे। और ये मिलेट्स हमारे न्यूट्री क्रॉप्स हैं, ये बहुत पौष्टिक तत्व वाली हैं, इसमें कैल्शियम, आयरन, मैग्नीशियम ये भरपूर हैं। लेकिन दुर्भाग्य है कि ये हमारे भोजन का हिस्सा नहीं बन पाई है।
साथ में मैं आपको ये भी ऐड ऑन कराना चाहूँगा कि इस मिलेट्स के ऊपर कोई सेशन भी रखा गया है।
हाँ, मिलेट्स पे हमने एक या दो सेशन रखे हैं। एक सेशन तो ये रखा है कि मिलेट्स को हम कैसे आगे बढ़ाएं, क्योंकि मिलेट्स का एरिया कम हो रहा है। ज्वार का एरिया अगर हम देखें तो करीबन 30 से 35% रह गया है और अब इसको हमें पूरा वापस ले जाना है तो बहुत प्रयास करने पड़ेंगे। बाजरा में लोग ग्रो करते हैं, लेकिन बाजरा में चूँकि किसानों को पूरा पैसा नहीं मिल पाता तो वो भी कई बार बाजरा की फसल भी कम ले रहे हैं और ये इसका असर जो सीधा पड़ रहा है वो हमारे स्वास्थ्य पे पड़ रहा है। तो इस सेशन में ये एक चर्चा है कि इन मिलेट्स की एक उपज कैसे बढ़ाई जाए और दूसरी इसकी गुणवत्ता है, इसको दूसरे हमारे जो धान हैं उनके साथ मिला के कैसे बढ़ा दें। मान लीजिए बिस्किट बनाया, बिस्किट में पर्ल मिलेट भी डाल दिया, रागी भी डाल दी और जो कोडो वगैरह ये डाल दी, तो इससे न्यूट्रिशन भी बढ़ेगा। और दूसरा जो सबसे बड़ा फ़ायदा होगा कि ये जो हमारी क्रॉप्स हैं, माने मिलेट्स और मिलेट, ये क्लाइमेट रिजिलियंट है। कम पानी हो उसमें चल जाती है, कम खाद वगैरह हो उसमें भी अच्छा दे पाती हैं। तो क्यों ना हम इसको प्रमोट करें। साथ ही में इसके द्वारा जो उत्पाद बन सकते हैं जो कि न्यूट्रिशन रिच, पौष्टिक तत्व युक्त, जैसे बिस्किट है, रवा है, इडली मिक्स है, कई तरह के बन सकते हैं। खुशी की बात है कि आज करीबन 300 स्टार्टअप इस एरिया में हो चुके हैं और 300 में कहूँगा ये तो बहुत कम है। राजस्थान जो सबसे ज़्यादा पर्ल मिलेट ग्रो करता है वहाँ पर बहुत ही कम है, पर्ल मिलेट का उत्पादन भी ज़्यादा है। तो ये संभावना है कि हम कर सकते हैं। तो हमने इस सेशन में कुछ वैज्ञानिकों को, कुछ जो लोग इसमें प्रोसेसिंग और वैल्यू एडिशन पे काम करते हैं उनको भी बुलाया है। एकेडमिक जो साइंटिस्ट हैं उनको भी बुलाया है और प्राइवेट इंडस्ट्री से, माइक्रो को, नेस्ले से, हिंदुस्तान यूनिलीवर से भी लोगों को बुलाया है ताकि बैठ करके ये चर्चा करें कि हम किस प्रकार से साधारण व्यक्ति के खाने में मिलेट्स को कैसे इजाफा करें।
बिल्कुल,
ये जिस दिन हो जाएगा मैं समझता हूँ कि किसानों की समृद्धि भी होगी। समृद्धि तो एक बात रही, लेकिन उन जो हमारे जनता है उनका स्वास्थ्य सुधरेगा।
बिल्कुल ठीक बात आपने बिल्कुल ये कही, क्योंकि स्वास्थ्य के लिए आज के टाइम में मिलेट्स बहुत ज़्यादा ज़रूरी है। जिस तरह का हमारा खान पान हो गया है, इतनी मिलावट की चीज़ें, मिलेट्स में मुझे नहीं लगता कोई कुछ मिलावट कर पाएगा।
हाँ नहीं, आप बात सही है। मिलावट तो एक ऐसा है कि लोग अपने फायदे के लिए करते हैं, हाँ? लेकिन मिलेट्स में कुछ होगा नहीं,
हो नहीं पाएगा।
जिस एरिया में ग्रो करते हैं वहाँ लोग खाते भी हैं आज, लेकिन वहाँ भी प्रॉब्लम ये हो रही है कि जो यंग बच्चे हैं वो कम खाना पसंद कर रहे हैं। ये भी एक बहुत ज़रूरी है कि उन बच्चों का रुझान वापस कैसे लाए पारंपरिक फूड पर।
एक चीज़ ये भी है सर कि मिलेट्स में आम तौर पे जो हम खाना खाते हैं, फास्ट फूड वगैरह खाते हैं, वो हम चटोरे होने के चक्कर में ज़्यादा खा जाते हैं उल्टा सीधा। लेकिन मिलेट्स में एक लिमिटेड है कि हम ज़्यादा खा ही नहीं सकते।
हाँ, ये बात बिल्कुल सही। लेकिन एक जो दूसरा फायदा है मिलेट्स का, इसमें क्रूड फाइबर बहुत होता है
जी।
और क्रूड फाइबर आप दूसरे जितने फास्ट फूड हैं उसमें क्रूड फाइबर ना के बराबर है। तो वो हमारे को इंडाइजेशन की प्रॉब्लम है, कई सारी गैस ट्रबल से दूसरी ट्रबल्स होती हैं। तो वो सब मिलेट्स खाने से नहीं होंगे।
नहीं,
और एक अच्छी बात जो आज हो रही है कि पिछले 1 साल में जो सरकार ने प्रयास किए हैं उससे अवेयरनेस बढ़ गई है।
नहीं, ये बात तो सर मानने वाली बात है कि प्रधानमंत्री जी बहुत ज्यादा जोर दे रहे हैं इस पे।
उसके अवेयरनेस की वजह से आज बच्चे जो है युवा वर्ग वो बाजार में जाते हैं तो देखते हैं कि प्रोडक्ट किसमें मिलेट्स है।
मिलेट्स,
और इसी वजह से नेस्ले कई सारी कंपनियों ने मिलेट्स के बिस्किट बनाना शुरू कर दिया, जो नई बात है।
जी, जी।
और मैं समझता हूँ ये एक अच्छा प्रयास है और आने वाले समय में बहुत ज़्यादा प्रोडक्ट्स आएंगे आपके इडली मिक्स में, सारे मिलेट्स मिक्स करके आएंगे। और हर रीजन में, क्योंकि हमारे यहाँ भिन्न भिन्न भात हैं, आप साउथ में चले जाओ तो खाना दूसरा है, नॉर्थ में चले एक दूसरा है, उन सब में हम मिलेट्स को कैसे शामिल करें ये बहुत ज़रूरी बात है। और खुशी की बात है कि जो हमारे वैज्ञानिक हैं उन्होंने जो प्रयास किया है उसमें ये संभव है कि हम मिलेट्स को हर खाने में मिला सकें। दूसरा जो मेरे हिसाब से गवर्नमेंट ने और अच्छा प्रयास किया है कि सेना के खाने में 10 से 15% मिलेट को ज़रूरी कर दिया। तो इससे मांग बढ़ेगी तो किसानों को ज़्यादा पैसा मिलेगा। अगर उनको ज़्यादा पैसा मिला तो वो निश्चित रूप से मिलेट्स को आगे बढ़ाएंगे और ये हमारे फ्यूचर के लिए बहुत अच्छा होगा। और इसी दिशा में हम लोग अभी एक और जो इस संगोष्ठी में विचार कर रहे हैं कि जो नई तकनीक आई है, आपने सुना होगा कि जीन एडिटिंग की बहुत बात चल रही है
जी।
जीएमओ तो हमने बंद कर दिए, जीएमओ के लिए तो काफ़ी अपोजिशन है। तो जेनेटिकली मॉडिफाइड जो है बायोटेक्नोलॉजी प्रोडक्ट हैं वो केवल बीटी कॉटन के अलावा कुछ किया नहीं है। अब जीन एडिटेड वेराइटीज़ आ गई हैं। मैं सोचता हूँ कि किसानों के पास अगले 2 साल में पहुँच जाएगी। इसकी वजह से अगले 4-5 साल में और भी ऐसी वैरायटी आएंगी जिनमें आपको खाद की बिल्कुल ज़रूरत ना हो, शायद आपको कोई केमिकल वगैरह छिड़कने की भी ज़रूरत नहीं हो। तो इससे हम हमारे जो प्रोडक्ट हैं वो कई सारे ऑर्गेनिक प्रोडक्ट दे पाएंगे जनता को, वो भी एक बहुत बड़ा फायदा होगा। कुल मिला के मैं समझता हूँ कि ये जो हमारी इंट्रैक्शन है इसमें चूँकि वैज्ञानिक यूएसए से भी आए हैं, कनाडा से भी आए हैं, यूके से आए हैं और अपने अपने एरिया में काम कर रहे हैं। और 2 लेक्चर हमने ऑनलाइन भी किए हैं, जो नहीं आ पा रहे हैं वो भी वर्ल्ड लीडर हैं, उनकी जब बात सुनेंगे, चर्चा होगी तो निश्चित रूप से ये आगे बढ़ेगा। एक और जो हमारा प्रयास हमारे सोसाइटी की तरफ से हो रहा है वो ये है कि हम नए बच्चों को ट्रेनिंग दें। तो हम साल में 2 ट्रेनिंग प्रोग्राम करते हैं नई टेक्नोलॉजी में। अभी हैदराबाद में हमने प्रोग्राम किए थे जीन एडिटिंग पे ट्रेनिंग देने, इसमें प्राइवेट सेक्टर से भी आए, वैज्ञानिक भी आए, साइंटिस्ट भी आए। ऐसे प्रोग्राम हम सब जगह करेंगे। साथ ही हमने ये निश्चित किया है कि लोगों में अवेयरनेस कम है, प्रौद्योगिकी के बारे में, जैव प्रौद्योगिकी के बारे में अवेयरनेस कम है। तो हम स्कूलों में लेक्चर्स, हमारे जो वैज्ञानिक हैं अलग अलग, जैसे मान लीजिए कलकत्ता हो गया, बेंगलोर हो गया, उदयपुर हो गया, इन सब एरिया में हम लेक्चर्स तैयार करेंगे। साथ ही वहाँ पे जो टीचर्स हैं जो ये बायोलॉजी पढ़ाते हैं, चूँकि उनकी ट्रेनिंग जो हुई है उनकी जैव प्रौद्योगिकी में नहीं हुई, लेकिन उनके सिलेबस में 10+2 के सिलेबस में जैव प्रौद्योगिकी का बहुत बड़ा हिस्सा है जी। अब वो किताब को देख के पढ़ाते हैं, उन्होंने किया नहीं है, हम उनको सिखा रहे हैं कि ये कैसे किया जाता है। जी, जी, जी। तो इससे उनका आत्मविश्वास बढ़ेगा, वो बच्चों को अच्छी ट्रेनिंग दे पाएंगे। और दूसरी जो भ्रांतियाँ इस समय में लोग फैला रहे हैं, मैं समझता हूँ वो भ्रांतियाँ भी नहीं फैला पाएंगी। जी, जी, जी। बहुत बहुत धन्यवाद सर आपकी।
Showed very clearly that the transgenic lines are free from the diseases, and it is very clearly showing that we could functionally validate the action of this particular gene, which was later on also validated by using RNAi approach. Now, what is the use of mapping and cloning of this particular gene in crop improvement program, which we showed very clearly in marker assisted breeding along with Dr. A.K. Singh's group in genetics IRI, New Delhi and different parts of the country. So marker assisted breeding, it's a very commonly used term wherein markers are used for interrogation of gene of interest from different species to their recipient 1, and these markers should be closely linked to the gene of interest. And here I would like to show that this particular linkage map here, which we developed by using map-based cloning as well as characterization of the gene, 2 DNA markers linking to the gene of interest, TRS26 and TRS33, and also gene-based markers were developed. If gene-based markers are there, then and you amplify it from the segregating lines, and you get a single band, as we showed in this particular gel picture. It shows that the gene is there, and if the gene is present in heterozygous form, then you can see variable pattern of the gene. So for doing that, we crossed it up with Pusa Vastupivan at IRI New Delhi with Dr. A.K. Singh's group and developed multi-location evaluation trials by using segregating populations. And it is also showed very clearly that we have grown region-specific varieties in the country, and a country-wide program was started to transfer PI54 gene in more than 40 varieties of rice. So it shows that the gene PI54 is effective to all the strains of Magna Proteoraceae present in different parts of the country, and this gene has already been transferred in more than 40 varieties of rice in India and abroad by public and private funded institutions. If you see overall scenario of National Agriculture Research System, we have successfully demonstrated in the country that more than 87 varieties of different crops have already been developed by using marker-assisted selection, and most of the varieties, 43, are reported in case of rice, and it itself tells us that rice is a very well studied crop as far as genetics and genomics is concerned. We could also see that now countrywide all these 87 varieties have also been improved for more than 24 traits, different traits, and most of these traits are controlled by single genes. But in spite of the fact all these efforts during these 20-25 years, if we see, we have large scope of improving this whole program, because in India we work on 85 crops, but still 7 crops are being taken care in case of marker-assisted selection. Rest of the crops need to be worked and need to develop more DNA markers and use in marker-assisted selection. The final topic which I would like to cover here is genome editing, about which we are talking since morning, but we say that genome editing is nothing but it's a precision mutagenesis tool, which was developed by these 2 ladies who got Nobel Prize in Chemistry in 2020, Jennifer and also Mueller. And this is one of the important technologies which we have now in biotechnologies, and luckily we are very fortunate to have that SDN1 and SDN2 types of these mutations have also been cleared by Government of India from any regulations. So if we are developing on SDN1 type and SDN2 type of mutations using CRISPR-Cas, then readily we can take this material to the field. And I'm happy to share that our colleagues at IRI and also Indian Institute of Rice Research, Hyderabad, have successfully used these genome editing approach, SDN1 mutants, and these 2 lines, MTU1010 and BPT5204, have been improved for high yield, drought, salt tolerance, and these 2 lines are now under field testing under AICRP trials, and we are hopeful that next year these 2 lines would be released for commercial cultivation after clearing from all the regulations. Similarly, if we see the status of different crop varieties which is going on in the country, and various institutions of DBT, ICAR or CSIR, these are working on different crops like rice, and also on different traits like high yield, drought, thermosensitive, sterility, aroma, high iron and zinc, sheath blight resistance, disease resistance, and also brown spot diseases. And timeline is very clearly defined when these varieties can be released in the field, and also their testing lines has also been defined. So more than 21 efforts are going on in the country at different institutions of DBT, CSIR or ICAR on different crops, and 10 crops are being taken care, which includes rice, mustard, wheat, tobacco, cotton, tomato, banana, tea, cannabis, and bidania. If we see now, new program has already been started by Indian Council of Agriculture Research under National Fund program with a total outlay of 7.87 lakhs. And in this, 10 institutes are involved in developing genome edited lines in case of mustard, soybean, chickpea, pigeon pea, rice, and sugarcane. And in this particular program, if you see, we have given more emphasis now towards oilseed and pulses besides rice and wheat. I am happy to share that Government of India has approved a mega project on genome editing of crops and traits in Indian Council of Agriculture Research, and 310 crore rupees have been earmarked to the genome editing of cereals, pulses, fiber crops, oilseeds, sugar crops, and tobacco. More than 24 crops we are taking up in this 5-year program, and for different traits which are agriculturally important. And it is being now discussed at various platforms, and final proposal is being made. Similarly, in genome editing of horticulture crops, 120 crore rupees have already been sanctioned for working on 15 crops, and all these programs are now being discussed at various institutions and also are underway at different stages of its development. Genome editing not only provides a golden opportunity to handle or manipulate single gene of interest, but it also provides an opportunity to look at those traits which are domestication difficult. Particularly for domestication of novel crops through multiplex genome editing is one of the exciting area of research. And if we see, this is an example of de novo domestication of wild species through genome editing. It's a new concept in which genome editing for multiple traits can be done, particularly for abiotic stress tolerance, disease resistance, nutrient quality, yield component, plant architecture, and agronomical adaptability. And with limited generations of yield trials, we can modify or we can improve wild species into the cultivated and commercial lines. So this approach has to be taken care in future in various crops in India and abroad. So looking at all these advancements in biotechnological approaches, what would be the way forward as far as national agriculture research system is concerned? We see the genetic gain in major crop is only about 1%, which needs to be increased to 2.4% for meeting global food demand by 2050. This demand can be met by application of pre-breeding, genomic selection, speed breeding, and genome editing technologies for genetic enhancement of crops. Pulses and oilseed production must be enhanced to meet the domestic demand. Application of pre-breeding, genomic assisted breeding, and frontier sciences for genetic enhancement and crop improvement needs more emphasis. Development of novel crop production, protection, and processing technologies for enhancing crop production in the realm of global climate change. Application of new technologies like AI, IoT, sensor, and big data analytics for phenotyping of large population and precision breeding is required. Frontier sciences such as genomics generated huge amount of genomic data. Deciphering the phenotypes from the genome sequence of different environments and pyramiding phenotypes to obtain designer crops are major challenges for the coming decades. We should also give more emphasis for quality breeder seed production until as we do emphasize this particular area, these all new technologies cannot be forwarded to the farming community. I'm sure that with this presentation I could make clear what we are doing at national levels and what are our future priorities. And I'm sure that all plant scientists sitting here in this gathering will definitely take note of this and will work in these areas. So with these words, I would like to thank you all for your patient listening. Dhanyavad.
Thank you very much, Dr. Sharma. If there is one pressing question, time is short, I would certainly like to take it.
So, we know there is a issue genome edited plant for the patent. So do you see in future like US will allow to use this tool without patent issue, or we should look alternative method of genome editing?
Yeah, there are two ways of doing this. One is that we are discussing with the US partners also. Government of India has made a team of scientists, including Director IRA Dr. AKC. They are discussing patent issues with the developers. Number two is naturally we have to develop our own tools.
Thank you. Thank you very much once again. Let us give him a big hand. And we will now move to the next speaker, who is Dr. A.K. Singh. He is director at ICAR and has been leading the program in rice breeding, genomics and genetics for last several years. He has addressed several problems which have come to him in the form of improvement of rice as a crop. So Dr. A.K. Singh from IRI.
Thank you, Chair. After an excellent presentation by Dr. T.R. Sharma on the basic and strategic research, I would like to take you through some of the translation research that we have done at IRI. My presentation is focused on three crops, primarily rice, maize, and wheat. So if we look at rice, and especially in case of basmati rice, the traditional varieties of basmati rice, they are pretty tall, prone to lodging, photoperiod sensitive, and they take about 160 days to mature, and yields are very low, about 2.5 tons per hectare. Realizing this limitation and also the fact that basmati is grown in a specific geography where we have limitation of the area, we needed to improve the productivity, and that's where in late 60s, Dr. Swaminathan, unfortunately, is no more with us. A program was initiated to combine the quality of traditional basmati in high-yielding background. The first class was made in 1968, and that led to development of first dwarf variety, Pusa Basmati 1, in 1989. If you look at the progress that has happened through conventional breeding, those days we didn't have marker, we didn't know the genes, genomic regions involved in these traits. But from 1930, when you look at the grain length on cooking of Basmati 370, a traditional Basmati variety, and present day variety like Pusa Basmati 1509, the length on cooking is significantly increased, almost double. You get about 1 inch length of rice. That means you get more cooked rice per unit rice that you cook. You can serve many plates, you can eat less rice, and you can feel that your belly is full. Even diabetes can afford to do that. Pusa Basmati was the first landmark variety, and then came 1121 in 2003, which revolutionized the basmati rice production and export. 2013, we came out with Pusa Basmati 1509, a short duration basmati variety which took only 100 days in field, about 20 days in nursery. So seed to seed 120 days and produced about 7 tons of paddy, bringing a revolutionary change and also diversifying cropping system. After harvesting rice, one could go for peas, one could go for potato, one could go for sunflower, and this led to diversification from rice cropping system. Today in Punjab alone, almost 5 lakh hectare area is planted to these early varieties, followed by alternative cropping system. The overall impact of this has been that duration has been reduced from 160 days to 120 days in present day variety as compared to traditional varieties, and the productivity has improved from 2 tons per hectare to almost 7 tons per hectare. This Caesar-like curve graph is very difficult to find in plant breeding. Generally, when we increase duration, we increase yield. If you reduce duration, yield decreases. We were able to break that unholy alliance, and we were able to develop varieties which were early, yet produce higher yield. And this culminated into a tremendous export earning of basmati, which is currently almost $4.79 billion annually. $4.79 billion annually is the forex earning from export of basmati rice, and almost 95% of this coming from the variety developed at IRI. I would also like to acknowledge here the total volume exported is about 4.5 million tons, which earns about 4.8 billion US dollar. If you compare this with non-basmati segment, where we are exporting 17 million tons, and we are not bringing this much of money. So it's still growing in less area, bringing more foreign earning. And tomorrow, if a decision is to be taken as a policy, whether by exporting rice, we are exporting water, and what should we stop, then of course non-basmati export can be considered to be minimized. This also has happened primarily because of the investment from private sector. On the left-hand panel, you see the investment that happened by private sector in installing high-capacity mill, which could mill the grain length of 9 millimeter. Otherwise, Samma Musuri, Jaya, IR8, these are small varieties, even normal rollers, they can mill without breaking it. But a long grain of 8 to 9 millimeter was not possible to be milled. And the millers came forward, the exporters spent almost 500 crores in one processing plant. And number of such processing plants were developed. Companies like KRBL, Saithya Overseas, LT Overseas, and many. And the credit goes to this investment from private sector in the technology that led to milling and enhanced export value while the varieties were available. But the current day challenge with our variety that they are highly susceptible to biotic stresses, particularly bacterial blight, blast, brown plant hopper, bacane, seed blight, brown spot, name a disease that is there. And this has resulted into excessive use of pesticide in basmati cultivation, but in particular, and of course in rice in general. And this has also led to return of many export consignments from European Union, where the laws are very stringent. The tricyclozole, one of the fungicide which is used for controlling blast disease, is limit fixed is 0.01 ppm. That is 1 milligram of tricyclozole in 100 kilogram of rice which is detected. Then the consignments are returned, and that resulted into reduction of basmati rice export from India from about 5 lakh tons in 2016 to 2.5 lakh tons in 2020 from EU alone. And therefore, conscious of this fact, we initiated breeding program to develop varieties which are resistant to these biotic stresses, so that there is no need to spray pesticide, and if you don't spray, there is no question of residue. Using molecular breeding, which provides a very efficient tool, being precise, accurate, early stage selection can be practiced. It is the costly and cumbersome phenotyping process is avoided, linkage drag is minimized, and these are the major advantages of molecular breeding. Here is a field I am standing in Panipat district, and you can see the panicles that I am holding in the hands, they have drooped down from the neck because it is affected by a neck blast. Neck blast affects the neck of the panicle, and therefore the translocation of photosynthesis to the grain is badly affected. Even at advanced stage, if it happens when the grain is milky stage, all the panicles will dry and you will have nothing to harvest. And therefore, we initiated this program. Dr. Sharma led a project for combining the major blast-resistant genes, looking at the host-pathogen interaction. Our focus was to transfer these genes into major varieties so that we could develop blast-resistant varieties. Most of these genes were available in non-basmati sources, and when non-basmati donors were used, they spoiled the quality of basmati rice. And therefore, we made a program so that we could transfer all the major blast-resistant genes in one basmati background. And therefore, the genes like PI1, PI54, PETA, PI5, PI9, PI2, and PIB, 7 genes we started marker-assisted simultaneous with backcross breeding program, where recurrent parent was used, Pusa Basmati 1, and finally we were able to develop isogenic lines of PB1 carrying 1 gene, 2 genes, 3 genes, 4 genes, 5 genes, up to 7 gene pyramids. And these pyramids can be deployed as and when the existing combination is broken. So one of these lines, Pusa Basmati 1637, which carried PI9 gene, which comes originally from Rhiza minuta, was released for commercial cultivation, and this provides excellent resistance to both leaf blast as well as neck blast. Farmers are not required to spray pesticide. Likewise, several other varieties in last 3 years we have released. that includes Pusa Basmati 1718 that carries XA13, XA21, 1728 also carries the same 2 genes, and then 4 gene pyramids, which includes 2 genes for blight resistance and 2 genes for blast resistance, including PI2 and PI54, the gene that Dr. Sharma was mentioning, PI54. And these varieties have been provided to farmers excellent response from the farmers' field. You look at this crop, and this crop can be compared with, an 8-ton yield per hectare, and this is harvested. This year picture I have taken from the farmer's field, Pusa Basmati 1847, which has been harvested now, and 32 quintals per acre yield this farmer has realized. And the rate that they have achieved is about 3,700 per quintal. And this brings 1 acre, almost 1,20,000 rupees from rice, one cannot imagine. So this is the kind of change that has happened because of gene deployment strategy. The farmers' cost of cultivation has been reduced by 3,000 rupees per acre because that's the cost for pesticide and fungicide generally used. Bacani is a major problem that farmers are facing, and this is caused by fungus Ibbac physicorae, which produces gibberellic acid in the plants, and the plants grow tall and ultimately they dry. So we had a recombinant inbred line population from Pusa 1342, a resistant variety to Bacani, which we crossed with 1121. This real population was used for mapping a gene for Bacani resistance. 3 major QTLs have been mapped, and these QTLs through marker-assisted backcross breeding have now been transferred to 1509 and 1121. They will be available shortly to farmers. Another very important area where we are working is the development of herbicide-tolerant rice varieties. We know that transplanted rice requires a lot of water. To produce 1 kilogram of rice, you have to spend 3,000 liters of water. And if rice cultivation has to sustain in northwestern part of the country, particularly Punjab, Haryana, and western UP, where water table over a period of 30 years has declined by 30 meters from 1970 to 2020, if you see, that's the kind of depletion has happened in water table over 20 meters. So shifting from transplanted paddy to direct seeded rice is the only option for sustaining rice farming in this region. But direct seeded rice or DSR suffers heavily from weed infestation, and therefore herbicide tolerant variety will provide a key component of DSR farming. We have developed 2 varieties recently, Pusa Basmati 1985 and Pusa Basmati 1979, which are improved version of 1509 and 1121. And this carries a mutation of ALS, acetolactate synthase gene, which provides a broad range control. It's a broad spectrum herbicide, imidazolpine, which is sprayed at 18 days crop. All weeds are killed, but nothing happens to crop. So these varieties will become available to farmers for direct seeded rice cultivation. And this is a plot of DSR farming using herbicide-tolerant rice variety. As you can see, the first picture field looks absolutely clean. Herbicide has been applied after 18 days of direct seeding. The crop is sown by seed drill, just like wheat, using 5 kilogram seeds per acre. Excellent weed control, and the yield realized by this farmer is 27 quintals per acre in DSR, saving lot of water, lot of labor, because for transplanting we need 4,000 rupees per acre transplanting cost, and also less greenhouse gas emission, almost 35% cut in that. So all advantages in DSR, and this has to go in big way. Also, we have combined the drought tolerance through a major QTL from N22 airplant variety, that is DTY 1.1, and that drought tolerance in DSR variety will provide further boost. Salt tolerance is another major QTL for salinity, seedling stage salinity tolerance, which has been combined in Pusa Basmati 1509, and these versions are also available. We have also employed the molecular mapping and molecular breeding approaches for nutritional enhancement of rice. And to address this problem, we had a set of 192 diverse rice genotypes which were evaluated in multi-location trial. Phenotype with respect to iron and zinc content, and on this we found that one of the lines, Karapunel, which is a land race from Tamil Nadu, contained zinc in milled rice up to 49 ppm. Our current day zinc enriched varieties are at 24 ppm. Now we use this panel with 50K genotyping. We mapped this quite a few marker trait associations were identified in the panel, and they were validated using Karapunel as one parent, and that was crossed with BPT 5204. And this validation, about 3-4 tutorials have been validated in the biparental population. We are now transferring these to enhance the zinc level above 28 ppm in basmati rice varieties. Likewise, phytic acid is an anti-nutritional factor which reduces the bioavailability of iron and zinc. We had a mutant population through pilling approach, which we screened and we identified low phytic acid mutant, which have been mapped, and now we are transferring them to basmati varieties to enhance the availability of iron and zinc. Oil is another, because this conference addresses biochemistry and as well as biotechnological tools for crop improvement. You see, in India, we are producing 155 million tons of paddy. At 10% recovery, it gives about 15 million tons of rice bran, which has, the potential is about 3.1 million tons of rice bran oil at 20% recovery. But we are able to produce currently 0.9 million tons. So 2.2 million tons is untapped potential for rice bran oil, while we are importing edible oil worth 1,60,000 crores annually. One of the major problem in this is that the moment we do milling, the embryo is destroyed, that releases lipoxygenase and that acts on the fats and therefore rancidity develops. So soon after milling, bran has to be processed for extraction of oil, it cannot be stored. And therefore, through mutation, breeding and also screening germplasm, we were able to identify the LOX null mutants, which do not carry lipoxygenase functional allele. And this has been mapped, and now we are transferring this to basmati rice varieties so that the basmati bran oil, and in fact one company, India Gate Price, is branding basmati bran oil as Basmola, and it sells for 700 rupees a liter. You can, best of the best oil, you can think about 300 to 400 rupees a liter, but this basmati bran oil is 700 a liter because some of the basmati varieties, they carry null allele and they have no problem of this rancidity. Coming to another crop, maize, where our group is working very intensively, led by Dr. Firoz Hussain, and maize hybrids with quality protein incorporating OPEC2 gene, which enhances the lysine and tryptophan content by 3-fold. And these hybrids have been released for a specific ecology. 2 hybrids which are biofortified with lysine tryptophan have been released for countrywide cultivation. One major gene which has been identified in China for this lysine tryptophan enhancement is OPEC16. So far, OPEC2 was deployed in Indian breeding program, but when we combine OPEC2 and OPEC16 together, then there is 51% higher lysine over O2 alone and 43% higher tryptophan over O2 alone. So when we combine O2 and O16 together, lysine tryptophan content can be further increased. So we are working on this, and also the provitamin A gene combined with lysine and tryptophan. The first hybrid that combined these two traits, Pusa Ibbac UPM9, has been released for commercial cultivation. Sweet corn we take, but if you see from health point of view, sweet corn has nothing but sugar, and our group has now very successfully combined provitamin A and quality protein maize, that is high lysine, high tryptophan plus provitamin A in sweet corn background. So while you are enjoying sweet corn, you can be also conscious of the fact that you have high nutrition. Bioavailability of provitamin A in human system, we have studied using Kako2 cell line in association with National Institute of Nutrition, and we find that some of these biofortified hybrids, they are able to meet 52% of recommended dietary allowance. One hybrid, the other hybrid 64% of recommended dietary allowances if we take 200 gram of provitamin A enriched maize. So this can be a great source for provitamin A enrichment. If you look at the global work done on biofortification, and India finds prominently a place in the global map where marker-assisted breeding for quality protein maize has been done. This recent review by Prasanna. And India is the country where we have got almost 25 hybrids which are released with provitamin A, more than any other country. The nutritional value of this biofortified maize, particularly with provitamin A, has been studied also using animal model, and particularly in case of poultry, we know that 75% of maize produced in the country goes for poultry feed. So through poultry route, we can have a better health if we provide a provitamin enriched maize and QPM maize to poultry as a feed, the health of poultry and the egg content of provitamin A increases tenfold. So this data clearly indicates, and also provitamin A being antioxidant potential, the aflatoxin production is less in these materials when this is to be used as feed after processing. Also, we have biofortified along with QPM, provitamin A, vitamin E, and alpha tocopherol has got less vitamin E activity. So, a mutant allele of BTE4, when incorporated, it converts alpha tocopherol into the gamma tocopherol into alpha tocopherol, and alpha tocopherol has got higher vitamin E activity. So with this, we get very high, almost 21.6 ppm vitamin E and provitamin A and lysine and tryptophan also 3-4 times. So these are the multiple trait biofortified maize hybrids which have been developed. And also to enhance the availability of iron and zinc, the low phytic acid mutant 1, LPA1 and LPA2, these are the two natural mutations which are available from the donor germplasm that we have deployed into our hybrid, so that bioavailability of available iron and zinc can be substantially improved. Waxy maize, if you go to northeastern region, the local varieties, they contain almost 100% amylopectin. They do not have amylose, and they are used for making porridge and various preparations. Current day hybrids are not in that quality range, and therefore for that target region we have developed hybrids which are sticky by incorporating waxy 1 locus, and this becomes extremely important. And one most important thing here is that their recovery of bioethanol from waxy maize is much higher. When we compared the number of hybrids available with our group, we found that the waxy maize hybrids, they have got about 15% bioethanol recovery, which is as good as rice. And this is where interest was shown by UP Distillers Association. They came forward, they have signed MOU with IRI, and they are funding our program to develop maize hybrids which can be used for bioethanol. We know that government has taken a decision not to allow broken rice for bioethanol production, and also government has targeted by 2025 to have 25% blending, 20% blending of bioethanol into petrol. And this would require about 1,000 crore liters of bioethanol. So unless we revisit our research program, crop diversification, this bioethanol has to come primarily from maize and sugar. And these two crops, either by conventional breeding, molecular breeding, or through genome editing, will find a major research focus in time to come. Low glycemic index maize as a breakfast food, because this has been a concern in our group for quite some time, and I have been challenging my scientists that morning breakfast cereal people take cornflakes. Usually wherever you go in hotels and Western counters, you find that item, but those who are diabetic cannot afford to take it. And therefore, if we can reduce the amylose content and also the resistant starch, so we have been able to now increase the resistant starch amount to 20.9%, and that reduces the glycemic index of these maize varieties to 44% as compared to that of 81% in normal maize. So cornflakes prepared from this would be really good for as a breakfast cereal. We know that haploid inducer stocks have been used for production of double haploid lines, which expedited the breeding program. And our lab in genetics division, Dr. K.R. Sarkar, who worked for his PhD with Co, was the first person to develop haploid inducer lines in 1960s. But the haploid induction frequency was very low, about 1%, 2%. And then temperate germplasm of the inducer stocks were developed with higher frequency. They were not good for Indian condition. Our group has now been able to tropicalize those inducer stocks, and Indian backgrounds. Now we have got haploid inducer lines, which we are using for development of double haploids in all our breeding program to expedite the breeding process and for quick fixation of zygosity. In wheat, one of our leading variety, SD2967, which was at one point of time grown at 10 million hectare, later became susceptible to leaf rust, yellow rust, and we have incorporated genes Trinacria for leaf rust and yellow rust both, and the improved version 3406 has been released recently, and this year we have given 1 kilogram packet to a large number of farmers, so hopefully there will be good impact in years to come. Likewise, another variety, SD2932, which was popular in central India, it became susceptible to leaf rust, which has also been improved for leaf resistance and improved version given to farmers. So large number of varieties. We have got marker assisted program, molecular breeding program supported by ICR, led by Dr. T.R. Sharma, Dr. E.K. Adva. And this program has resulted rich dividend in terms of number of varieties in different crops which have come up in last about 7-8 years. This is another variety of which it combines the QTL for drought tolerance and terminal heat tolerance. The mapping work was done, and after mapping, this variety has been released through marker assisted selection. So way forward, Dr. Sharma eloquently mentioned all those points, and my points are no different. Integration of speed breeding for further acceleration, and we are coming out with 2 speed breeding facility, though it's taking little time at Indian Institute of Pulse Research and at IRI. Gene editing, and of course creating novel alleles through gene editing, pre-breeding, and modifying plant architecture for productivity, packing more number of plants per area. For example, in case of maize, there is a lot of opportunity globally. People are going for very dwarf corn, where the planting density can be increased to double at the current level of say 70,000-80,000 per acre to 1,20,000. So if that is achieved, then probably productivity will double. And then of course bringing together the biotic, abiotic stress tolerance, nutritional quality, and higher yield. These are the ultimate goal in our breeding program. I would like to thank my colleague, Dr. Firoz, Dr. Gopala, Salint, PK Singh, Vignesh, and Rajkumar, Ranjit, Harita, Niharika, and Hari Krishna, who have contributed to this presentation. Thank you very much.
Thank you very much. Any pressing question? You can raise hand or press the button of your mic. Okay, maybe I could ask them questions. You see, you had raised in the morning a point that when these marker resistive breeding is being used to transfer the genes, it is being done in some of the varieties, which has a challenge of loss of diversity. So, are there concerted effort in our agriculture system to avoid such a scenario in coming years?
Yeah. So we have a very strong program which is supported by ICR and Bill and Melinda Gates Foundation, and that is focused on genomic selection. And genomic selection is one tool that can bring in higher seeding to yield by in fact focusing in the entire genomic region, not focusing on one or two specific loci which are introduced through marker assisted backcross breeding. And of course, in conventional breeding program, the breeders are making those diverse crosses. But of course, definitely, my concern was that when a new technology comes, there is a lot of emphasis on that technology. But all the outcome of that technology of molecular breeding, for example, so far we have released about 87 varieties through molecular marker assisted breeding in different crops. But most of these all these varieties are improvement of the existing variety, right? So we can protect the yield of that previous variety, which the genetic potential we can protect, but we cannot increase that genetic potential. So for that, I think more concerted efforts are required on genomic selection and also using more diverse genetic base in breeding programs.
Very fine. So, thank you once again. Dear colleagues, I'm sure you enjoyed these fascinating talks. It gave a cross-section of lot of activity which is going on in India on plant breeding, genetics and genomics assisted breeding also. Of course, you might have seen that each of the talk had been based on the experience of respective speaker. Like Dr. Sharma was talking about the blast resistance, and Dr. Singh emphasized various rice germ blasts which have been produced. Also, there was an interesting point which Dr. Sharma has brought out, and that was about domestication by genetic editing. This aspect has not been taken, but at least research papers in general are showing that there is a lot of potential, particularly with those crops which need to be domesticated and are waiting for that because of activity of one or other gene. So this is also an area which could be taken up further. I could go on talking, but everything has been talked by these two speakers. Thank you very much, therefore, once again. And
Thank you, sir, and our distinguished speakers for sharing expertise with us. Their insights have certainly enriched our understanding of plant biology. May I now request Professor Akhilesh Tyagi, sir, to felicitate our distinguished speakers with a token of appreciation. Thank you, sir. May I now request Professor S.L. Mehta, sir, to please come onto the stage to felicitate our honorable chair of this session.
दोनों रोल हमने आपको देखे, एक तरफ इतना टफ और एक तरफ इतना सॉफ्ट। ये दोनों मैनेज कैसे आप कर पाते हैं? आपने जो शुरुआत की नई तकनीकों के साथ आपने ये काम शुरू किया और आज इस उपलब्धि का
आपने एक ही प्रश्न में बहुत सारी मुश्किलें डाल दी मुझे और कैसे मैं जवाब दूँ, क्योंकि शब्द तो मेरे पास नहीं है इन सब चीज़ों के लिए सही मायने में। लेकिन सबसे पहले तो आपने पूछा कि ये दो जो चीज़ें मैंने दिखाई यहाँ पर, जो मुझे जितना पार्ट मिला मेरे इस प्रोग्राम में, तो एक तो टफ और दूसरा है सॉफ्ट। एक्चुअली जो सिक्के के दो पहलू है या जो दो चीज़ें हैं, जो जोड़ा है जिन जिन चीज़ों का, चाहे वो सूरज और चांद का कहिए, चाहे वो औरत और मर्द का कहिए, और इस तरह के आप सैकड़ों उदाहरण देते चले जाएं जो द्वैत है। हम सब द्वैत हैं, क्योंकि अद्वैत एक ईश्वर है, वो ऊपर वाला हमारा जो मालिक है वो अद्वैत है। हम सब द्वैत है, जोड़े के साथ हैं। तो उनमें से जो पुरुष और प्रकृति का जो रूप है, वो एक पुरुष स्ट्रांग होता है और जो प्रकृति है वो सॉफ्ट होती है। तो उसमें जो ये दो भाव जो है, मन के दो भाव, ये दोनों भाव प्रबल रूप से हम सब लोगों के अंदर प्रदर्शित होते रहते हैं, झलकते रहते हैं हमारी भावनाओं से। तो उन्हीं दो भावनाओं के नाते वो दोनों काम मैंने किए। भवाई जो हमारा है, इसके बारे में मैं कहूँगा कि वो राजस्थान का एक बहुत ही पुराना परंपरागत, पता नहीं कब का, हजारों साल पहले हुआ हो, उसका इतिहास आज भी लिखा हुआ है उदयपुर लोक कला मंडल में। और वहाँ के जो संस्थापक थे देवीलाल जी सांभर, उन्होंने अपनी किताबों में इस तरह का जिक्र किया कि अजमेर जिले में केकड़ी तहसील के पास एक नागोड़ा गांव है, वहाँ एक नागाजी नाम के बुजुर्ग हुए और उन्होंने अपनी पत्नी के साथ जब वो शीतला माता के मंदिर गए, शीतला माता जो चिकन पॉक्स की देवी है, उनके यहाँ पर दर्शन करने गए, तो वहाँ उनके मन में आया कि माँ का तो इतना चमत्कार है, मैंने बहुत नाम सुना है इनका, और मैं क्यों ना इनसे कुछ मांगूं, क्योंकि मैं 55 साल का हो गया हूँ, बुढ़ापा बहुत करीब आ गया है, लेकिन मेरे को कोई बच्चा नहीं। हे माँ, आपका अगर चमत्कार और शक्ति है तो मुझे भी दिखाओ और मेरे को एक औलाद दो। तो माँ ने प्रसन्न होकर उनके मन की बात सुनी और उनको तुरंत अपने समय के अनुसार बच्चा हुआ। लेकिन उन्होंने ये भी कहा था उस वक्त मन्नत मांगते हुए कि हे माँ, मुझे बच्चा मिलेगा तो मैं आपके इस मंदिर में आके बहुत बड़ा उत्सव करूँगा। तो जब बच्चा हो गया और वो उसके उत्सव में लगे, बच्चे के उत्सव में घर में ही, उसके बाद उसके लालन पालन में लग गए, व्यस्त हो गए। 3 वर्ष का बच्चा हो गया, लेकिन माँ के मंदिर जाने का उनको फुर्सत नहीं मिला। तो शीतला माता के लिए बड़ा मशहूर है कि वो कुपित हो जाती है, रुष्ट हो जाती है, और चिकन पॉक्स की देवी है, फिर वो अपना पेट दिखाती है। तो माँ ने उसके अनुसार उस बच्चे को ऐसा चिकन पॉक्स हुआ कि वो मरणासन हो गया, अंतिम सांसें चलने लगी। रात के टाइम में दोनों पति पत्नी लेकर बैठे हुए थे, नागाजी जाट और उनकी पत्नी। अचानक माँ स्वप्न में आती है, आँख रखती है नागाजी की, तो कहती है कि हाँ भाई जाट, बहुत बड़ा उत्सव आके कर रहा था मेरे मंदिर में, तुमने तो एक पानी की लुटिया भी नहीं मेरे ऊपर डाली, और ये तो गलत बात है। मेरे कोप से, मेरे प्रकोप से मैं रुष्ट हुई हूँ तुमसे, और मेरे उससे ही ये तुम्हारे बच्चे की हालत हुई है। अगर इसको जिंदा देखना चाहते हो, खड़े हो जाओ, गांव सरपंच जी के पास जाओ, सारे गांव को इकट्ठा करो, गाजे बाजे लो, कुमार के घर जाओ, 7 कलश वहाँ से लो, फिर कुएं पर जाके शीतल जल से उन कलशों को भरो, 7 कलशों को, और सारे सर पे उठा के जल से भरे हुए कलश, और नाचते गाते बजाते सारे गांव के साथ मेरे मंदिर में आओ और एक एक कलश मेरी मूर्ति पर खींचो, तो मैं तुमसे प्रसन्न हो सकती हूँ, ये मेरी शर्त है। तुरंत बच्चे को माँ की गोद में पकड़ा के भागा नागाजी, और ये सारी चीज़ दोहराई, माँ के मंदिर तक पहुँचा, वो सब करते हुए जो माँ ने बताया, और वो माँ पर 7 कलश जैसे सींचे, बच्चे की आंखें खुल गई, उसकी सांसें चल गई, और माँ का चमत्कार हुआ, उनकी शक्ति के दर्शन हुए। और नागाजी ने तब से फिर वो त्याग दिया अपना घर संसार, गांव ही त्याग दिया, वही मंदिर में ही बैठ गए वो, और रोज सुबह उठते थे, 7 कलश लेते थे, कुएं पर जाते थे, जल के भरते थे, और वो जो गाजे बाजे वाले पहुँचते थे मंदिर में, वो उनको नचाते हुए कुएं से मंदिर तक लेके आते थे, और फिर माँ को रोज सींचते थे। और ये क्रम चला, ये होता रहा, उनकी देखा देख कई लोगों ने गौर किया, और ये होते होते आज ये पता नहीं सैकड़ों हजारों सालों बाद ये नाच विकसित होता 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 राजा महाराजाओं के, मुगल बादशाहों के, अकबर के, जो उस किताब में भी मिलता है कि इस तरह के मटके लेकर नाचने का जिक्र किया हुआ है।
थोड़ा सा करीब आ जाए।
तो इस तरह से करके, और हमारे राजस्थान में जो भरतनाट्यम में भी ये डांस होता है।
अब मैं आपके एक्ट पे एक बात पूछना चाहता हूँ
जी
कि जो आपका एक्ट देखा, वैसे तो पूरा एक्ट ही अमेजिंग था, बट जो लास्ट में आप तलवार पे और कांच के टुकड़ों पे जो खड़े हुए और उस एक्ट के एक्शन को देखकर आपको स्टैंडिंग ओवेशन मिला यहाँ पर और तालियों की गड़गड़ाहट कंटिन्यू रही, तो उस टाइम आपके मन में क्या ख्याल थे, किस तरह का आपके विचार थे, किस तरह की खुशी थी, उसके बारे में बताइए।
जी बिल्कुल, मैं इतना ही कहना चाहूँगा, ये तो बहुत अलग चीज़ है, इसके लिए भी शब्द नहीं है सही मायने में, क्योंकि दिल में उतरना और दिल में जाकर कोई दस्तक दी जाती है तो फिर उसकी हलचल तो दिखाई देती है। तो लोग उसी नाते खड़े होते हैं उस चीज़ को देखकर, और उसमें और भी कांच और तलवार तो है ही है, वो मेरा अभ्यास है जिनकी वजह से मैं चोट लगने से बचता हूँ हमेशा। लेकिन जो बैलेंस है एक ग्लास के ऊपर अलग अलग खुले खुले मटकों का, वो बैलेंस इतना ओरिजिनल है कि मैं हजारों प्रोग्राम कर चुका हूँ। हर प्रोग्राम से पहले मुझे डर लगता है, मैं इबादत पर बैठ जाता हूँ और उससे मांगता हूँ कि हे ऊपर वाले, हे ईश्वर, हे कन्हैया, तू आ और खुद संभाल मेरे इस बैलेंस को, मेरे बस का नहीं है। मैं सिर्फ एक माध्यम मात्र हूँ, वही नाचता है, मेरा कृष्ण नाचता है, वही बैलेंस बनाता है और वही ये सब करतब करता है मेरे पास आकर। मैं उनका अनन्य भक्त हूँ, मेरे कन्हैया का, बस। और कुछ मुझे लेना देना नहीं दुनियादारी से। बाकी तो लोगों में जो देखेंगे वैसी वो उनके अंदर उसकी प्रतिक्रिया है। और मेरा एक बहुत बड़ा सौभाग्य है कि आज इस इंस्टीट्यूट में इतने महान लोगों के सामने मैं ये अपना प्रदर्शन कर पाया। मैं बहुत बहुत धन्यवाद करूंगा कि ये कॉन्फ्रेंस हुई जो आपके सामने है, और इस बहाने मुझे यहाँ पर बुलाया और मेहता साहब मिले यहाँ पर, और आप सब लोगों के दर्शन हुए। डॉक्टर अनिल सिंह जी और अर्चना मैम का मैं विशेष धन्यवाद देना चाहूँगा, क्योंकि उन्होंने हमें शुरू से लेकर अब तक कोऑर्डिनेट किया और इस पूरे प्रोग्राम में बहुत मेहनत की कि हम इसको इतना छोटा कॉम्पैक्ट करके, डांसेस को छोटा कर करके एक डॉट टाइम दिया था कि 60 मिनट से ऊपर नहीं होना चाहिए। मैंने 60 मिनट 1 सेकंड का ये पूरा टोटल सीक्वेंस तैयार किया, बस वो 1 सेकंड मेरी तरफ से था एक्स्ट्रा। जी
चलिए, थैंक यू सर, थैंक यू।